is through fundamental research to understand the mechanisms of the disease as the basis for development of new effective treatments. Margaret Turner Warwick was Professor of Thoracic Medicine and the Dean of the National Heart and Lung Institute. She was renowned internationally as the leading clinical academic in her specialty in the UK. She made many major contributions to several aspects of respiratory disease, but her enduring legacy is her research in pulmonary fibrosis. I'm delighted to welcome here today members of Margaret's family, her daughter Lynn, and her grandchildren Tabitha and Michael, who are continuing the family tradition in medicine. I should also like to welcome Robbie Rayne. Robbie is chairman of the Rayne Foundation, of which Margaret was a trustee for many years. His wish to make a major donation to commemorate Margaret stimulated the idea for the centre, and most importantly, provided the initial funding. I should also like to thank Richard Reed and the trustees of the National Heart and Lung Foundation, who provided a similarly generous donation. Finally, I would like to welcome Professor Jonathan Weber, Dean of the Faculty of Medicine, who provided critical support for the appointment of a chair to lead the centre at a financially uncertain time for the faculty during COVID, you may recall. We are very fortunate to have appointed Professor Gisli Jenkins to the Margaret Turner Warwick Chair in Thoracic Medicine to lead the centre. Gisli was previously Professor of Experimental Medicine at Nottingham University. He has kindly agreed to give the 2022 Margaret Turner Warwick Lecture entitled Curing the Incurable. Professor Jenkins is an international leader in the field of pulmonary fibrosis. His research focuses on the biological basis for its development, translating this understanding into improved outcome for patients. He is the principal investigator of a number of longitudinal observational studies, including the PROFILE study, the INJUSTIS study, and the UK Interstitial Lung Disease Post COVID ILD study. In 2020, he was awarded the ERS Gold Medal in Interstitial Lung Disease. It gives me great pleasure to invite Gisli Jenkins to give this year's Margaret Turner Warwick Lecture entitled Curing the Incurable. Gisli.
Thank you. So thank you so much for that kind introduction. And so, yeah, I will be giving the talk, Curing the Incurable. And as many of you know, this talk was meant to be given by Martin Kolb, so I've adapted his title a little bit. And that's the importance of the alveoli in lung fibrosis. And Margaret Turner Warwick knew this long ago. Indeed, she did coin the term cryptogenic fibrosing alveolitis. So these are my disclosures. And just to start off, I'd like to say that everything in this talk and all the work that I have done has been motivated and inspired by talking with patients with pulmonary fibrosis through interactions in the clinic and through collaboration with patient groups such as Action for Pulmonary Fibrosis. And this has been formalized in a James Lind Alliance NIHR collaboration with Action for Pulmonary Fibrosis. And the initial results, the preliminary results, which were put together by Lara Fabry, who's a clinical fellow in the group, demonstrate the priorities for patients. And one of them is finding a cure, which many of us thought would be beyond what was possible. But their persistence in demanding that we as a community find a cure for pulmonary fibrosis is what has led me to this point. So the strategic plan for the Margaret Turner Warwick Centre for Fibrosing Lung Disease is to find a cure for pulmonary fibrosis. So how are we going to go about this? So I'm going to start by describing the problem that is IPF. And then we need to identify people at risk of IPF, preferably before symptoms and irreversible scarring has occurred. And then determine which of these people are at risk of progressive disease and focus on this group. And then ideally, we will prevent progression completely, or even better, promote resolution of fibrosis. So this is idiopathic pulmonary fibrosis. This is the clinical scenario. Usually there is a 2 to 3 year history of progressive shortness of breath and exertion and a dry, non-productive cough. Many patients have been diagnosed with congestive cardiac failure or COPD prior to their diagnosis. Ex-smoking is a big factor. So they're not current smokers. Only 12% of patients with IPF are current smokers. 75% are ex-smokers and gave up many, many years ago. So this is not recent ex-smokers. In contrast, it's about 50-50 for COPD. There's a history often of working in dusty environments such as building and manual trades. And we're recognizing now more the importance of family history. And I'll come on to that later. It's a disease of advancing age. There is a predominance in male sex. And again, importantly, it's associated with comorbidities such as systemic hypertension, type 2 diabetes, and ischemic heart disease. And this will become apparent, the importance for this will become apparent as we go forward. And on examination, we find digital clubbing, and you'll see this picture that I obtained from Margaret Turner-Warwick's thesis, demonstrating the key features of digital clubbing. So you lose the angle between the nail fold and the nail, and you get increased blood flow. And I'll come on to that later. And when you listen to the chest, you find Velcro inspiratory crepitations. And importantly, it's a progressive disease. So these are CT scans taken from the native lung of people who've had a single lung transplant. And as you can see in the non-transplanted lung, the small amount of fibrosis at time of transplant, and these are people who are ill enough to get transplanted, progresses over about 4 or 5 years. And without the transplant, these patients would undoubtedly have died of their fibrosis. And the incidence of IPF is rising. So it went from 1968 to 2008, it increased 6-fold. And in a subsequent analysis commissioned by the British Lung Foundation, we found that there was a doubling of the incidence of fibrosis in the 10 years from 2008. And there is no signs of it slowing down. And my prediction is that it will actually increase further. And again, you'll find out why during the course of this talk. And if you're unlucky enough to get pulmonary fibrosis, the survival is poor. The 5-year survival is worse than most cancers, not shown on this graph, but it's about the same as esophageal or gastric cancer, with only lung and pancreatic cancer having a worse prognosis. And the median survival, and these are data from the PROFILE study, which I'll talk about in more detail, is about 3 to 3.5 years. But it's not just about mortality. It's important to remember that from the point of diagnosis until the end of life, that the condition progresses. There's progressive disability with increasing oxygen requirements, and this is a picture from a patient support group. And you'll see at the back, patients may be ambulant, they may require ambulatory oxygen, but as you see towards the front of this photograph, patients are often in wheelchairs, requiring 24-hour oxygen towards the end of their life. So it's from the point of diagnosis onwards, there's a lot of disability. And it's an incurable disease. And a bit like the runaway train, at the moment, once it starts, nothing can stop it. And that's what we're trying to change. So I take this clip from, again, from Margaret Turner-Warwick's thesis. And this is wonderful. So she said, when a subject has been dominated by speculation for greater than 100 years, so now closer to 200 years, there is an important place for recorded anatomical observation. And what she also said in there is that finger clubbing was likely the explanation for finger clubbing was likely to come from biochemical and physiological studies. And I think this is true for all of pulmonary fibrosis. And this picture is of Margaret with Jeff Laurent, who is a biochemist and who is my PhD mentor, who helped supervise my PhD. And of note, we now think that finger clubbing is due to increased VEGF and PDGF. So Margaret Turner Warwick was right. So this is the anatomy of normal lung, and the cartoon shows the key features. So you have a very nice alveolus. It's a beautiful structure made up of type 1 and type 2 epithelial cells. This is the capillary, made up of endothelial cells. This blue stuff, this is the interstitial matrix, which is the glue that sticks it all together. And there's not a lot else in there. There's the odd alveolar macrophage, the interstitial dendritic cell, and the interstitial fibroblast. And that's just shown here. And importantly, the basement membrane on which the type 1 and type 2 alveolar epithelial cells sit is shared with the endothelial cells where it joins the vascular bed. And it's a very thin structure. And it's easily damaged by things like oxygen-free radicals, inhaled smokes and dust, and even before COVID, it was damaged by viruses such as flu and RSV. And then from the circulation, it can be damaged by immune-mediated injury. And the outcome when this happens is the same. You get fibrosis. And this micrograph is taken from a patient who had pulmonary fibrosis. And as you can see, the very fine, delicate structure is completely obliterated. You've got this thick interstitial matrix. This is fibronectin and collagen-rich matrix. There is inflammatory cells and apoptotic cells within the alveolar lumen. So you see here, activated macrophages, neutrophils, lymphocytes, apoptosis. And the basement membrane is destroyed. So this basement membrane is destroyed, and that's important. Again, you'll see why later, because it allows interaction between the bronchialized epithelium and the proliferating fibroblasts, which secrete the fibronectin and the collagen. So what our goal is to understand the journey from normal to fibrotic lung. And what we know is it takes a long time. So that's the first thing to say. As I said, 75% of patients are ex-smokers, but they gave up 30 years ago. We know from asbestos and asbestosis, the exposure is often many years before the disease, which I think is an important thing when we consider the consequence of COVID. And I'll talk more about that later. And so then from exposure, in early adult life, you develop these things called interstitial lung abnormalities, which are asymptomatic, and we wouldn't know they existed if we hadn't started doing screening studies, because we haven't normally CT'd people who don't have symptoms. And then, as I said, you get 2 to 3 years of symptoms, cough, shortness of breath. People often just think they're unfit, because apparently, getting older is synonymous with being less fit, but I just don't think that's true. And then the part of the disease that we've actually studied is this bit here. So this is idiopathic pulmonary fibrosis, and most of the research we've been doing up until this point has focused on this end stage of the process. So we need to look earlier. And the screening studies have taught us a lot. Initially, this is data from Rachel Putnam and colleagues published 5 years ago now. And using a whole bunch of different cohort studies, they realized that patients without any lung disease, or in the case of COPD-G and COPD, have these reticular abnormalities, which we call interstitial lung abnormalities. And if they have those, they do worse. They have a higher risk of mortality than patients without, or people without interstitial lung abnormalities. But it takes them a lot longer to reach that point. So it's not 3 years, it's more like 10. But look at the figures. 7.5% of screened populations have an interstitial lung abnormality. If you look at family members, so this is the first-degree relative of someone who has sporadic IPF, 25% of these people have an interstitial abnormality. So no symptoms, but the radiology suggests that there's something awry. So back-of-the-envelope calculations, based on current estimates, we've got about 1 million people with interstitial lung abnormalities in the UK. So it's a lot of people with potential fibrosis. So how do we know if they're going to get worse? Well, actually, lung function is pretty good. These are data which Fazzy Khan, who's a clinical fellow in Nottingham, are just preparing. And what it shows, and these are placebo arms from all the clinical trials that have been published in IPF over the last 10 to 20 years. And actually, a change in FVC over 3 months of 2.5%. So these are not big numbers. Is predictive of increased mortality. Similarly, the same can be seen in disease progression, which is a further worsening of FVC or death within 12 months. And again, only a 2.5% change in FVC in 3 months is predictive of poor outcome. We need to bear in mind that these are prospective clinical trials, so the spirometry is done at a very, very high standard, and not necessarily generalizable across all spirometric assessments. And you'll notice that if there's a decrease of 5%, the risk is increased. But we don't have to, so you can measure short-term changes, small changes in FVC, and it's not great news. And one of the things we wanted to do was to look at biomarkers. So this is the PROFILE study. This is a study that I set up with Toby Marr, and he was at the Brompton, and we set this up 12, 13 years ago now. And it was the first prospective longitudinal observational study of incident IPF. So these are patients who had not had IPF forever, but had been diagnosed within 6 months. And we've followed them up over 3 years, measuring various bits and bobs, including quality of life questionnaires, physiology, and biochemistry. And the first thing we looked at was matrix neoepitopes. So matrix neoepitopes measure matrix turnover. So in a nutshell, when matrix is put down, it's remodeled, and it's remodeled by matrix metalloproteases, and the ones that we focused on in the initial analysis were MMP2 and 9, and they cleave up the matrix that's deposited. And so every matrix molecule is cleaved, giving a fragment, a neoepitope. So collagen 1 becomes collagen 1M, collagen 3, collagen 3M, and so on and so forth. And basically any molecule which is a substrate for MMP2 and 9 will generate a fragment. And what we found, so green are healthy controls, blue are stable patients with stable pulmonary fibrosis over the course of a year, and red are patients with pulmonary fibrosis that progresses. And what we found was that for a range of these matrix neoepitopes, so this is by glycan, collagen 1, collagen 3, cleaved by AD and TS, collagen 5 and 6, CRP, cleaved by a matrix metalloprotease, and vinculin. And these are the ones that came up in the initial analysis. And what you can see is that in progressive disease, levels of these neoepitopes were higher than in stable disease, and they were mostly higher than healthy controls. But the important thing was the change over time. And when we dichotomized the results into patients who had stable or rising levels of these neoepitopes, what we found, particularly for CRPM and C6M, was that patients with rising levels of matrix neoepitopes had a worse survival, which was consistent with progressive matrix turnover being associated with worse outcomes. We then looked at an unbiased proteomic approach to see whether we could discover other novel biomarkers of pulmonary fibrosis. So we stratified by disease versus healthy, progression versus stable, and change over 3 months and mortality. And what was not surprising was that surfactant protein D, a marker of epithelial injury, and MMP7, another marker of epithelial injury, were able to distinguish disease from healthy. And we've seen this in early ILAs. These things rise early. What did surprise us a bit was that the best predictive progression was CA19-9, carbohydrate antigen 19-9, which many of you will be familiar with as a pancreatic tumor marker. And even more surprising was CA125, classically an ovarian tumor marker, bearing in mind that it was highest in men who don't have ovaries. So it's not coming from the ovaries, but rising levels were associated with increased mortality. So this was in a discovery set, so we replicated this. We also wanted to check where the CA125 and CA99 were coming from. And interestingly, that's also known as mucin 16. So you see it in normal bronchial epithelium. Carbohydrate 99 it attaches to carbohydrates, which are a component of mucus. Again, you see it in normal bronchial epithelium, but it's hugely upregulated in the honeycomb cysts of patients with fibrosis. So you see it in the epithelium, and they're just churning out mucus, which gets stuck in the honeycomb cysts, and these label that. And then when the assumption is when the disease gets worse, it spills over into the blood. And so in the replication analysis, we focused just on SPD, MMP7, CA99, CA125. And what you'll see in progressive disease, higher levels of SPD than in stable disease, but it doesn't change much over 3 months. MMP7, not quite as distinctive, but the same pattern, higher levels in progressive versus stable. CA99 was quite remarkable because this, where the confidence intervals just touch, is 37 international units. This was done in a clinical chemistry lab at the City Hospital in Nottingham. So this is it was the standard assay that you would all use. And the upper level of normal is 37. So if it was high, they had progressive disease. If it was normal, they had stable disease. CA125 is slightly more subtle in that all patients had normal levels, pretty much. But if it rose over 3 months, then they had progressive disease. And again, when we dichotomized to those who had rising levels of these biomarkers over 3 months, patients with rising levels of CA125 had an increased mortality, so 2.5 increased risk of mortality, which is substantial. In fact, that was the highest we've seen in any of our biomarkers. Didn't change much with SPD and MMP7, which we think reflects the time it takes for this stuff to change. So what happens if we add biomarkers to lung function, and if we look at lung function together? So these are some data produced by Hanan Feinberg, who's here in the audience today. And so initially, there's a whole load of prep that went into this, but basically using artificial intelligence and self-organizing maps, you can cluster patients into 4 types based on lung function. So patients who have progressive disease over the course of 3 years, those who have an initial decline and either sadly pass away or stabilize out. And then you have those who improve a bit over the first year, and then they gradually decline. And then you have a group of patients who are stable for the whole 3 years. And the more dots, the more people in that cluster. And so this is the biggest cluster. This sort of, as I call it, the Newtonian cluster, is fairly common, about a third of patients. And 14% of patients have stable disease. And I think we've all seen this group of patients in clinic, the patient who doesn't seem to get worse. So is there anything about these clusters that we can learn biochemically? Well, bottom line is that this group tend to be younger, tend to be never smokers. These have more current smokers. Bear in mind, there aren't many current smokers at all. And they have a high level of SPD. So epithelial injury is driving this. And just a little bit on the MMP7, mainly because it's been measured by so many people. So we've been able to do a meta-analysis on it, as all the other biomarkers haven't been replicated enough to do a meta-analysis. But what we see is that high levels of MMP7, again, indicate progression. So you can measure this at baseline, and it will predict progressive disease. And that's with every, that's the increased risk with every standard deviation above the mean of MMP7. And similarly, mortality, so the higher your MMP7, the higher your risk of mortality with pulmonary fibrosis. And so the final thing about biomarkers, and one of the reasons we really wanted to do this in the first place was we wanted to see if we could get a treatment response. The trouble with measuring lung function in IPF is it goes down regardless of what you do, with the exception of the Newtonian cluster, where it goes up regardless of what you do. So you don't change the trajectory with antifibrotics. You just alter the amplitude. So you progress more slowly, but it's very difficult to measure that in an individual patient. But with CA125, and we assessed all of these biomarkers in a study with BI, the INMARK study, so they used nintedanib. And the protocol was pretty simple. The first 3 months were placebo controlled, and one group got nintedanib, one group got placebo, and then after 3 months, everyone gets nintedanib. And what you see with CA125 is that within 4 weeks of starting nintedanib, CA125 drops, and then it stays flat. Then in the placebo arm, it doesn't change until you add nintedanib, and then it drops, and then it stays the same. So it's a potential theranostic biomarker, as in if you take nintedanib and your CA125 drops, that's good news, relatively speaking. So I put it to you, and this is Martin's slide, that IPF pathogenesis begins at the epithelium. Actually, when I first saw the title of this slide, I thought it was the end. So IPF pathogenesis begins at the epithelium, the end. Pretty short talk. But actually, I think what he means is that the pathogenesis begins at the epithelial end of lung development. And pulmonary hypertension starts at the endothelial end. But I'm not going to talk about pulmonary hypertension today. But I'm going to talk about the epithelium in more detail. And one of the key fibrogenic molecules is transforming growth factor beta-1. And it does everything that you would want a profibrotic molecule to do. It prevents alveolar repair, promotes matrix deposition through a bunch of different mechanisms. But it's kept in latent form by association with the latent TGF-beta binding peptide, which then binds the small latent complex through the latency-associated protein, which has an RGD domain. This is really important. If you remember nothing else for the rest of the lecture, remember the RGD domain is important, because that binds integrins, and integrins activate this to release TGF-beta and allow it to become fibrotic, allow it to exert its fibrotic effects. And integrins are cell surface heterodimers. There are 18 alpha and 8 beta subunits. They heterodimerize to form 24 different combinations, and 8 of them contain an RGD binding domain, which means they can bind and activate TGF-beta. And we've shown that 5 of these can do this in vitro, and actually most of them can do it in vivo. And so what happened, or what this was the sort of hypothesis I was working on when I went to San Francisco 20 years ago nearly now, is that the epithelium is injured. That causes something to happen within the epithelial cell, which promotes cytoplasmic tug on the cytoplasmic domain of the integrin, which exerts pressure on the latent TGF-beta binding, the latent associated peptide of TGF-beta1, which allows it to be released from the latent complex, bind to its receptors, and then induce profibrotic signals. So we thought if we can find out the mechanism by which this happens, we've got a plethora of targets to try and inhibit. And so if we used LPA, which is a G protein coupled receptor agonist, what happens, this is an epithelial, it doesn't really show so well, but these are epithelial cells, very relaxed. And this is the beta-6 integrin. I don't know if you can see that because of the lighting, but there's beta-6 integrin around the cell surface. And this is the actin cytoskeleton. And if you stimulate this with LPA, it changes the shape. And you get pronounced stress fibers, and you get relocalization of the integrin to focal contacts. And we believe it's this physical restructuring of the integrin that activates TGF-beta. And there are a number of different molecules that we've been shown to do that. So lysophosphatidic acid is one, but thrombin and S1P, which we've developed a recent interest in and are collaborating with Jodie Rosenblatt from King's on looking at this, activate their respective G protein coupled receptors to activate G-alpha Q11, Rho A, and Rho kinase. So we mapped this out in pretty great detail in vitro. Was it happening in vivo, and what was the effect? So we knocked out the G-alpha Q11 signaling pathway in epithelial cells of mice, which was quite a Herculean effort undertaken by us and John. It took there's a lot of crossing and breeding, and it was hard work. Well, it was for Alison. And what you can see at 2 weeks, the lungs are essentially normal postnatally. But at 6 weeks, you see these inflammatory lesions, which just get progressively worse over time, and you can see that the air spaces are increasing. So there's inflammation and emphysema, and following the principles of Margaret and Warwick, we assessed this anatomically by measuring mean linear intercept, and it gets increased in the knockouts. Physiologically, by measuring lung function in the mice, so total lung capacity was increased, residual volume was increased, consistent with emphysema, and biochemically, by measuring MMP2, 9, and 12, all of which were increased. It wasn't developmental emphysema, it was matrix metalloprotease-induced emphysema, which we see when the TGF-beta pathway is inhibited. And so we looked at TGF-beta activation in the lungs of these knockout mice, and it was reduced. So there's reduced TGF-beta1 in lung protein, and using macrophages as a reporter cell, we were able to detect reduction in phosphorylation of SMAD2 in the macrophages. So knocking out GQ11 signaling in the epithelium only leads to reduced TGF-beta activation in the lung. And indeed, if we ventilated these mice, and this was a collaboration with Taka Masaka, what we found was that you got increased TGF-beta in wild-type mice, and this was reduced in knockout mice with high tidal volume ventilation. This is normal. So if you overventilate, if you have, it's the mechanism of acute lung injury in ITU, essentially. You overventilate, they get high levels of TGF-beta. You protect the mice from this when they don't have TGF-beta signaling in the epithelium. And measuring this physiologically, looking at resistance, they don't get the increased resistance you see when you develop acute lung injury. So again, knocking out the mechanotransduction pathway in the epithelium alone prevents a fibrotic response in ventilator acute lung injury. The downside is they get emphysema. We also have genetic evidence that this pathway is important. And this is work that I did with Louise Wayne and Richard Allen, a long-standing collaboration in Leicester. And so we did a GWAS a few years ago now. We identified that MUC5B and desmoplakin polymorphisms were associated with IPF, replicating what many people had shown before. But we identified a new gene, ACAP13, which was also associated with pulmonary fibrosis, and that's a ROGEF. ROGEFs are super important because they transmit signals from GQ11 to Rho. So that bit of the arrow, I'll come back to it in a cartoon later. But so that was genetic evidence that this pathway was important in IPF. And when we looked at EQTL, we found that this, the polymorphism, which was associated with this G to A change, led to increased levels of ACAP13 in normal lung. So this was, and these are 3 cohorts from Canada and Holland. And if they were homozygous for the mutant, they had highest levels of ACAP13 in normal lung. So we'd gone to great lengths to discover the normal physiology of epithelial response to injury and stretch in normal lung. So lysophosphatidic acid thrombin, S1P, through their G protein coupled receptors, signal through GQ11. This is amplified by ACAP13 to Rho A, then to ROC, pulls on the integrin, activates TGF-beta, sets off a SMAD3 signal, which leads to integrin suppression of macrophages. If you remove this brake, the macrophages go bananas and mice and people get emphysema. So that's all well known. So how does this go wrong in people with pulmonary fibrosis? Well, the first thing we thought was, well, ACAP13, if it's amplifying, should increase, there should be an increase in beta-6 and TGF-beta signaling pathways. And certainly in IPF, there's an increase in ACAP13, a big increase. Again, it's found in the epithelium of the bronchi and in type 1 and type 2 cells, and it goes up in the epithelium of patients with pulmonary fibrosis. We also see lots of it in lymphoid follicles. We have no idea what it's doing in the lymphoid follicles, but it's there, and we don't see those in normal lung. And indeed, in mice, and these are mice that have received bleomycin, we're measuring the integrin by a PET signal, so the brighter the yellow, the higher the integrin. And you see mice with bleomycin get lots of integrin. If you block that, the integrin goes down, and this is just quantification of that. Nothing surprising there, but it was a new way to measure integrin expression globally, because the only other way to do it is via histology, which is not great in patients. But if you do get lung sections for patients with fibrosis, you also see an increase in beta-6. Not in all patients, but in most patients, but some patients have lower levels, and this is associated with prognosis, so the higher the level of the beta-6 integrin, the worse the prognosis of a patient with pulmonary fibrosis. And if you inhibit the integrin, and this is using a small molecular inhibitor from GSK, you can reduce integrin levels in the lung, again measured by SPECT scanning. You can, and this is quantified here, and you can reduce the TGF-beta activation, again using macrophage phosphorylation of SMAD2 as our readout. So a very nice concentration-dependent inhibition using an inhibitor of integrins. In mice, you can reduce hydroxyproline, and you can reduce matrix neoepitopes, as I mentioned before, these things that we measure in the serum. So you can prevent TGF-beta and fibrosis in a mouse by inhibiting beta-6 integrins. And in lung slices, we showed that you could inhibit TGF-beta release and pSMAD2 in the lung slice using the inhibitor. So biochemical support that this might be helpful. And what we and others have found and shown for a number of years is that there's an autocrine loop. So if you activate TGF-beta, you increase beta-6. This is flow cytometry, looking at beta-6 levels on the cell surface, and this is just quantified. So more integrin leads to more TGF-beta, leads to more integrin. So a feedback loop. So where does that go wrong? So this is work done by Amanda Tatler. She used promoter deletion constructs. We found this huge enhancer element in the promoter of the beta-6 integrin. And that enhancer region contained a number of interesting transcription factor binding sites, including glucocorticoid and progesterone receptors. But the one we were interested in, because there'd been the work from colon cancer showing it was regulating integrins, was the ALK1 ETS1 binding site. And there are two of those in the promoter, and if you knock them both out, this is what happens. So this is an ALK1 knockout mice, and they have high levels of integrin at baseline, and if you give them bleomycin, it goes up compared with wild type. They also get worse fibrosis. So if you knock out ALK1 in a mouse, it gets worse fibrosis, consistent with its function as a repressor of integrin function. Patients with pulmonary fibrosis have high levels of beta-6 integrin. I've shown you that before. Normal lung doesn't have much by immunohistochemistry. You can measure it by PET scan. But there's lots of ALK1 in normal lung. And in the regions of this fibrotic lung, you see the ALK1 is completely absent. So there is evidence that ALK1 is important in patients. So if ALK1 as a repressor was important, we wondered if they would get spontaneous age-related fibrosis, and they did. So this is a birefringent analysis of serous red, and that's collagen. You can see it here. So there's not a lot of it, and if you look at the interstitium of the knockouts, it's thicker, and this was measured. So the mean alveolar wall thickness, it's not a huge effect, but it was thicker than the wild type, and there was a little bit more collagen. So this isn't causing rip-roaring spontaneous fibrosis, but there is a slight increase, and it's mainly collagen 3. And interestingly, these mice also developed hepatosteatosis, which is the liver precursor lesion to liver fibrosis, showing that some mechanisms are shared across organs, which will not come as a huge surprise to anyone, but these fibrogenic pathways span different organs, and that will become important later on. So this is our current working hypothesis for how this pathway sits in And this is just one pathway of a number, but this is a pretty important pathway. So G protein coupled signaling is amplified by a mutant in ACAP13, a SNP in ACAP13, which pushes everything down this pathway to activate more TGF-beta via integrins. You lose ALK1, so you don't repress your integrin expression, so you get lots and lots of integrins on the epithelial cell surface. If the basement membrane is disrupted, that allows the epithelial cell to interact with the fibroblast, and these fibroblasts secrete matrix, and that's what causes fibrosis. So there's nothing necessarily intrinsically wrong with the fibroblast, but they're being led astray by their neighborly epithelial cells. So what about COVID-19? Okay, the reason I got interested in COVID-19 will become obviously apparent, but it's the exemplar example of viral-induced epithelial injury. It's caused by the SARS-CoV-2 virus. When I counted yesterday, it's about 250 million infections, cases rather, so double that for infections, about 5 million deaths. Unvaccinated, the hospitalization rate's 8%. At the last count, 575,000, give or take, admissions, 435,000 discharges. And the data suggests that patients who are discharged from hospital with COVID have breathing problems, and a substantial proportion may get long-term breathing complications. Severity of disease is worse in older people, male sex, comorbidities, hypertension, type 2 diabetes, and ischemic heart disease. Does that sound familiar? Yeah, these are exactly the same risk factors as you have for idiopathic pulmonary fibrosis. So this is where I got really excited. Remember I said, remember the RGD domain. It's really important. Well, within the SARS-CoV-2 receptor binding domain, that's this bit here, there is an RGD domain, which means, in theory at least, it can bind integrins. So SARS can bind integrins in theory. The thing is, it's a cryptic domain, so it's this little red bit in here, which means at resting state, it won't bind integrins because it can't, there's physical interaction. But when it binds ACE2, it reveals the RGD binding domain, and that can then interact with integrins. I was almost as excited when I discovered this as I was when I discovered that ACAP13 was a rogue F. And those are the two bits of information I've shared with my children. They were completely disinterested. So we did some binding assays, and these were done by Jess Carver, who's in the audience. And ACE2 binds spike protein, no surprise, in a very nice concentration-dependent manner. But interestingly, the alpha-v beta-6 integrin and the alpha-v beta-3 integrin, which you find on endothelial cells, that's all I'm going to say about it, but blood vessels may be important, will also bind spike protein. And it's abolished by EDTA, which means that it's an RGD-dependent mechanism. So at least in a solid phase binding assay, there is evidence that spike can bind integrins. What does this mean for viral cell entry? And these are data that we produced with Tom Peacock, who worked with Wendy Barkley. What you can see is that SARS has a KGD domain, which also binds integrins. So these are pseudoviruses with expressing luciferase, and so you can measure viral entry by the amount of luciferase. And this is what ACE2 does on its own. So if you overexpress ACE2 in cells that don't have any ACE2, this is how much viral entry you get. And you get nothing if you only overexpress the integrin. As I said, it's a cryptic domain, so if it isn't binding ACE2, it shouldn't bind integrins, and it doesn't really get in. However, if you overexpress ACE2, beta-3 integrin, or beta-6 integrin, you get an increased amount of viral entry by these pseudovirus assays, suggesting that there may be some physiological effect of these integrin binding SARS. And we see from these cytochemistry studies, so white is where you have spike protein and ACE2. You only get spike binding to integrin where it's also co-localized with ACE2 on the epithelium. So why is this important? Well, we know that in fibrosis, there's a big increase in beta-6 integrin. We've seen that. I've shown you that many times already. There isn't a lot of ACE2 in normal lung. 1.5% of AT2 cells express ACE2. It's increased a little bit in the small airways of patients with fibrosis, and that's quantified down here, which may be the reason that if you have an interstitial lung disease and you get COVID, things are worse. And this was a Herculean study done in collaboration with the ISARIC-4C consortium and every ILD unit in the country. And we knocked this up in about 6 weeks. I mean, it was quite remarkable, the enthusiasm of the community to get this done. And what we found was that patients with pulmonary fibrosis have almost twofold increased risk of death if they're hospitalized with COVID. This was in the first wave. And this was worse if they were obese and worse if they had impaired lung function, which is consistent with what we would expect. And so the big question everyone asks is, will COVID-19 lead to pulmonary fibrosis? Well, it can. These are CT scans from Cape Point and Nottingham, and you can see that pre-COVID, there's nice normal lung with about 22 centimeters from the diaphragm to the apex. 3 weeks after COVID, lots of inflammation, some traction change. 6 weeks, the inflammation's gone, but there's a lot of fibrosis. So it can happen, but this is not by any means universal, thankfully. And indeed, in this systematic review done by Lara, again in the audience, this is all patients who'd had a CT at the time of analysis, including Chinese studies. So if you have a CT and you've got COVID, we're pretty good, actually, in terms of deciding who should get a CT. Nearly 100% of people with COVID who have a CT requested by their clinician have some evidence of inflammation, and this gets better over time. So we've got up to 6 months. If they have fibrosis on their scan, it's a lot harder. There's a lot more variability, a lot more heterogeneity in these studies, which may be down to descriptions, et cetera, et cetera. But the bottom line is it doesn't shift much on the CT scan, which is what we're finding with lung function, actually, that if they have a reduced gas transport, it doesn't shift much over time. So are there any sort of reasons for this? So I just highlight this. This is the genomic study led by Kenny Bailey. And they found DPP9. You say, well, what's so interesting about DPP9? That's in 2 slides' time. In a recent update, they also found that ATP11A was important. So why do we get excited? Well, this is our GWAS for IPF causation. And you find that DPP9 is a causal variant for IPF, as is ATP11A. So there's shared genetic risk factors for IPF and severe COVID. But the biggest risk factor is MUC5B. So what happens if you have MUC5B and you get COVID? Completely different story. This is fascinating. And so this is a Mendelian randomization, and a lot of people have done this on a smaller scale. But what you find is if you have MUC5B, you have less chance of having symptomatic illness, less chance of being hospitalized, and substantially less chance of ending up on ITU. So it's protective. And so we did a leave one out analysis, which is great. It does exactly what it says on the tin. You look at the risk of severe COVID with all the alleles, and you just leave one out, and you go down the list. So this is all 16 risk alleles for IPF, and you leave one out. And it doesn't shift. You've got a marginal increased risk, which is non-significant, so these are your 95% confidence intervals, until you leave out MUC5B. And then all other risk alleles for IPF increase your risk of severe COVID, whereas MUC5B brings it back to normal, suggesting that there are 2 distinct endotypes. MUC5B is very much restricted to the lung, it's mucin 5B. All these others are systemic genes. ACAP13 found in all cells in the body, DPP9, all cells in the body. Which brings me to multimorbidity. So these are the 4 horsemen of the apocalypse: COVID, alcohol, smoking, and obesity. And you'll see they all cause multi-organ problems, all cause multi-organ fibrosis. And these are data produced by Ian Stewart, again, who's in the audience. And we found that if you have one fibrotic organ, in this case lung, only a third, just over a third of people haven't got a second organ involved. 40% have 2 organs, 17% 3 organs, and a small number have 4 organs fibrosed, based on loose definitions. And again, liver and renal, largely the same. So fibrosis is a process which affects all organs. We tend only to focus on the organ that we're interested in. So what is the effect on IPF if you have other organs involved? If you have liver disease or chronic kidney disease, your risk of mortality increases, which is not a huge surprise. Your MUC5B actually reduces your risk of poor outcomes, which other people have shown. We've always been slightly befuddled by that, but it's a well-recognized phenotype. ACAP13 SPD doesn't really change your mortality, and there's a slight hint that hypertension and diabetes may be affecting it. The hypertension risk is really interesting if you stratify by genotype. So MUC5B is a risk modifier only if you have hypertension. So if you don't have hypertension, it doesn't matter what allele of MUC5B you have. If you have hypertension, MUC5B is very protective. We think this is revealing a backdoor confounder. So the genes associated with hypertension are causing severe disease, not that MUC5B is protective. So what are the genes that cause hypertension, and are they related to IPF? Well, and this was, again, a huge surprise, published in, actually, Louise Wayne was a co-author on this, and I said, why didn't you tell me that TGF-beta was important in hypertension? And she said, oh, yeah, it just slipped my mind. I didn't really think about hypertension and fibrosis. But yeah, so TGF-beta signaling is a major signaling pathway in systemic hypertension. So I think we should be staging fibrosis. If this were cancer, and there are many similarities biochemically, biologically, and clinically, we would stage, the first thing you do if you have a tumor in any organ, you do a PET scan. Where else has it gone? We should do that in fibrosis, and we can with MRI. It's really good at detecting fibrosis across organs. There's a little bit of work to be done in the lung, but lung elastography is coming. And it's dead easy in the rest of the organs. And we know this is important because in short telomere syndrome, the progressive fibrosis knocks off organ after organ after organ, and ultimately pulmonary fibrosis is prognosis defining because the lungs are so important, for want of a better word. So we really need to get the fibrosis before it hits the lung. And now there are ample ways that we can do that. And then we need to remap IPF. I mean, we know if we've learned one thing from, we've learned lots of things from the COVID pandemic, but the recovery study, the remap CAP study, these platform studies are the way forward. We've got loads of targets. I'll just highlight some of them. Many of these are cheap, metformin, easily available ACE inhibitors. Treprostinil is being used, but loads of targets. We just need to rattle through the targets. Every patient who we see with IPF should be entered into a clinical trial in the same way that every patient who ended up in hospital with COVID was randomized in recovery and remap CAP. So I'm going to finish here and just say that IPF is common and serious, and I think because of COVID it's likely to become more common. It's genetic and environmental risk factors for IPF that prime the alveolus for fibrotic response to injury. So in a way, the injury is less important than the host response. So if you've got the wrong genes and you get COVID, fibrosis may ensue. Alveolar injury leads to G protein-coupled signaling, which activates TGF-beta by the alpha-v beta-6 integrin. And we have a suite of biomarkers that relate to prognosis, endotype, outcome, and we may be able to use these to develop predictive models and to guide therapy. There are a number of targetable mechanisms that we can investigate. So this is how we cure the incurable. We identify who will get the disease before they get the disease. We then stop them getting the disease. And we do that through influencing policy. And that's important. I haven't talked about that, but policy is important. So smoking cessation, pollution, climate change, etc. If we can't stop people getting the disease, we stop them from getting worse, or better still, we make it better. And that's where the experimental medicine comes in. And then we put everyone through a clinical trial. And we can't do it alone. And that's the one thing I've learned from a very early age. We need to do this collaboratively. And so I thank everyone who's been intimately involved in this work. So Joe Port, Amanda Tatler, and Chitra Joseph, who remained in Nottingham. Hanan, Alison, Ian, Lara, Sam, and Jess, who accompanied me from Nottingham. All the consortia, of which there are many, the UKILD Long COVID Consortium includes many of the people who've been involved and are listed here. The Demystify Consortium, which is another huge effort involving people from all these universities, all these charities. You see that all the major organisms involved are interested, pharma. The funders, of course, without whom none of this work could ever happen. And special thanks to the Rayne Foundation and the National Heart and Lung Foundation for setting up the Margaret Turner Warwick Centre for Fibrosing Lung Disease. It's a wonderful, wonderful thing to be working in the only centre for fibrosing lung disease in the UK. And of course, to Margaret Turner Warwick for being such an inspiration. So thank you, and I'll take any questions.
So thank you very much, Gisli, for a fantastic talk and how we're going to move forward. We can take questions from the floor, but also if you're listening online, then if you can type your questions into the chat, then Alison will read them out to us. So I'm going to start off, take chair's privilege. I was really interested in your aged elk mice, and I just wondered, had you looked, was there a sex bias in the development of disease, and have you kind of given them a second hit with a kind of injury model?
So yeah, so the blio was the first thing we did, and they get worse fibrosis. So yeah, it's an X-linked gene. So they were all male that we used. So we didn't look at the sex bias because elks only expressed in the X chromosome.
Rachel.
I'm going to try and organise my thoughts a bit. So, a lot of our efforts have always focused on the fibrogenic response, and a lot of pharma and a lot of what we did was looking at the fibrogenic, the fibrotic, fibroblast biology. I think what you're trying to convince us is we really need to pay attention to the epithelium because that's driving the fibrogenic response. So, my question is, do you think that there is a point in the disease course when it is the fibrotic response that takes over, and then the epithelium actually is no longer driving? There's this matrix that's stiff and its mechanical transduction pathways are disrupted, and then it is completely independent of the epithelium. And therefore there are potentially two phases to this, and if your paradigm of getting patients early, then really we need to get an epithelial biology focused therapeutic approach. But if we've got patients that are halfway down the course of their disease, we still have this issue that the fibrogenic response might be independently generated or feed-forwarding itself.
No, you're absolutely right. So we see this with TGF-beta activation. So the epithelium will drive signaling to the fibroblast, but you can activate TGF-beta, as you know well, through other integrins, so non-epithelial specific integrins. So the alpha V beta 6 integrin is only found on epithelium, not anywhere else. But alpha V beta 5 is found in mesenchyme, and alpha V beta 1 is found in mesenchyme. So it is possible that they can become autonomous. But when we looked in the lung slices, We were unable to completely inhibit TGF-beta activation by using alpha V beta 1 or alpha V beta 5. So it's possible that you can develop autonomous TGF-beta signaling in a fibroblast, but we didn't see that in a tissue slice. But clearly in other pathways, they could be driving autonomous fibrosis. Once the epithelium, as you say, once the train has left the station, all bets are off. And certainly, by the time you've got established fibrosis, the therapeutic approach targeting fibroblasts is probably the best approach, again, because the train has left the station. But if we really want to make a difference, pirfenidone and nintedanib are both effective, but they only slow it down. People still progress. People still, sadly, pass away. So we need to catch the train before it's left the station.
Peter.
So, Gisli, fantastic talk. So, Professor Warwick was interested in many different types of fibrosis, as I think that quote beautifully illustrates. Do you think there are big differences in the pathogenesis, or are the pathways that you described generic and apply to fibrosis of any cause?
Yeah, so, yes and no. So we have these discussions at MDT. I'm very much a pragmatist. I like to see things as fibrosis is, once you've got fibrosis, it doesn't really matter where it's come from. It's where it's going that's important. There are clearly different types of fibrosis, but I think it's less the etiology that's important than the host response, which will be determined genetically. So, fibrosis caused by chronic hypersensitivity pneumonitis, in a subgroup, they do just as badly as IPF, and we see this with the new sort of description of progressive pulmonary fibrosis phenotype. Once you've got progressive pulmonary fibrosis, regardless of etiology, whether it's connective tissue disease, sarcoid, asbestosis, they were all in there. The outcomes are bad. It may not be, there may be subtle differences, there may be a few months here or there in terms of overall prognosis, but actually they all progress. So once the fibrosis becomes autonomous, it doesn't matter where it's come from. So again, I learnt my analogies from JBT, but again, you go through trains, many ways to get to London on a train. You can try and block them one by one, but you maybe want to focus closer to the terminus.
Dodie.
Dodie. That was great. I guess what I'm wondering about, baked into this, it seems like senescence is at play, but I also find it really interesting that you have early smoking or early damage, and then there's this lag period. That's very confusing to me. And do people, if they continue to smoke, have less chance? I mean, it kind of
Counterintuitive. Yeah, so
prescribe cigarettes.
So this is what my patients say, I should have given up smoking. I shouldn't have given up smoking then. I say, no, you'd have died of lung cancer 10 years ago. So there is an advantage to giving up smoking. And the way I look at it is that whilst you're smoking, you're shortening your telomeres. So, you're essentially aging yourself, but not sufficient to cause disease. You need time after that to age you further. And that's the simplistic way I look at it. So you've just, you've chemically aged yourself, but you still need more time to develop fibrosis.
Could the smoking be upregulating MUC5B?
It's possible. I haven't looked. I don't know if anyone else has looked. We don't quite know why MUC5B causes fibrosis. It's the sort of the best explanation we've heard for why MUC5B causes fibrosis is that it allows the fibrogen to be in contact with the epithelium for longer. But there's very, very limited evidence for the mechanism of MUC5B-induced fibrosis. And there's not for want of trying. There's been a large number of studies which have just haven't nailed it yet.
Isn't it protective, did you say?
No, the MUC5B minor allele increases your risk of IPF, but is protective of severe COVID, which is completely different to all the risk alleles for IPF.
Justin.
Thank you for a wonderful talk. I was intrigued by your ideas for repurposing common drugs for Santon, metformin, given that we had huge numbers of people already taking these for many years, can we look back and see if there's any evidence that they were protected against it?
So people have done that. The problem is always confounding by indication. So you don't give people a drug unless you think it's going to work. And you know, there could be knock-on effects which you know, make it difficult for us to deconvolute that. But when people have looked, the things that seem to be protective are simvastatin. ACE inhibitors, not so much. Aspirin, not so much. But there's not... I suspect part of the problem is that there are different endotypes. There are different patterns of fibrosis. So I don't think everybody will have fibrosis of the lung, liver, kidney, and heart. So we probably need to target the right patients with the right modality.
Gisli, back to alpha beta-6. In multi-organ fibrosis, I'm trying to bring that theme back to your alpha beta-6 because that's epithelium restricted. And TGF-beta applies to all these other organs as perhaps the final fibrogenic factor. Then alpha beta-6 doesn't have a monopoly on all these other organs. What do you think may be the parallel mechanism that leads to TGF-beta activation that's autonomous?
Well, every organ, with the exception of the heart and the brain, and the blood vessels, has an epithelial surface. So certainly we see that alpha beta-6 integrin are important in liver fibrosis and kidney fibrosis. They're not as important as in the lung. There are other mechanisms, so kidney particularly, there are other mechanisms to activate TGF-beta, so plasmin, for example. But it's important in the liver. There are data, I don't think they're published yet, showing that inhibiting alpha beta-6 is beneficial in liver fibrosis. But you're right, in other organs, so heart, hypertension, it's probably not alpha beta-6 mediated, but again, there are other ways to activate TGF-beta, other integrins, other pathways. You know, it's a bit of a Pandora's box.
Jane?
Thank you, Gisli. That was really excellent. Thank you. You showed a slide about fibrosis in multiple organs, which was really interesting. And I think it just blinked and missed it, but you seem to be suggesting that where there was fibrosis in more than one organ, the lung was the last organ for fibrosis. Was that right?
Yes, that's the simple hypothesis. It's not proven or disproven yet, but it tends to be prognosis defining. So if you get fibrosis in the lung, your chances of surviving more than 5 years are limited, whereas if you get fibrosis in most other organs, your prognosis is much, much better. So it's probably for that reason that the lung is the final organ to fibrose.
Alison, do you have any online slide questions? At the back, sorry, I can't see.
Gisli, great talk. You mentioned about the RGD domain, which is shared by the coronavirus. Do you think that that could be harnessed as a vector with therapeutic intent for antagonising the fibrotic process at the level of the integrin?
Yes, and there are studies ongoing. I don't know if they've been, I know they were planning to get them through to CPAC. There is a biotech company that was making them. But I don't know whether it's progressed. I think actually the thought was it may be even more useful for preventing the lung injury than particularly preventing the uptake. I think most viral entry is mediated via the ACE2 molecule. It's a cofactor which may cause damage, but even if it isn't causing damage in the lung, just blocking integrins will affect vascular leak and acute lung injury. So they were definitely pursuing that. I don't know if it got beyond CPAC.
Okay. One more name, Jonathan. Go ahead.
Thank you. Lovely talk, really enjoyed that. You talked a lot about biomarkers being useful to identify patients at risk for disease progression. Is there any data that any of them give you a bit more pathogenic insight into the different endotypes that might respond differentially to the treatment options available or being developed? Is that useful?
Yeah, no, that's a great question. I have some data, it's very preliminary, but yeah, it does appear the matrix neoepitopes and the epithelial biomarkers segregate. So you can have a sort of matrix predominant. And now whether that comes back to Rachel's point that they're just more autonomous, we haven't seen a difference in prognosis yet. The epithelial biomarker, so CA125, for example, is if it goes up, it's just as bad for your prognosis as if it's a matrix neoepitope. They do seem to separate into distinct endotypes, which may be helpful for targeting, coming back to Justin's point about which drug class we use in which group. That's where we're trying to get to in terms of repurposing. So if you have an epithelial biomarker, use an epithelial target. If you have a matrix neoepitope, use a matrix target. That's exactly what we're thinking.
Jonathan.
You showed an extraordinary increase in prevalence over the last 20 years. So is that because susceptible people have stopped smoking?
It's a number of things. I suspect giving up, as I said, giving up smoking, I mean, you know, they would have died of something else earlier. So it may well be part of it. Smoking rates have dropped, which means people live longer, which means that other diseases come into play. So it may well be related to that. It's definitely not just ascertainment bias. Again, we can look at when CT was introduced. It didn't. There was a slight peak, but the trajectory overall didn't change. It's a real increase. If you look at other fibrotic diseases, for example, I mean, asbestos is doing a similar thing, a slightly different time scale, but sarcoid, flat, chronic HP, not so much. So it's not just ascertainment. So it's probably aging. It's probably, you know, other, you know, smoking cessation, other risk factors, you know, pollution levels. Interesting to find out.
Fantastic.
I think you said there was something like 14% of your patients have static disease. My first question is, when you first see a patient, can you, the expert, tell which category they're going to be in?
No.
Okay, the second question is, do you think that out there's a great deal more people with the static disease who essentially don't think they're symptomatic because they're elderly, they don't take a lot of exercise, but they may be a substantial number of people with static fibrosis?
So potentially yes. I mean, so the first thing to say, it's static lung function. Actually, they still sadly die, not as quickly as patients who have progressive loss of lung function. So there's something else at play. But certainly, if you think about interstitial lung abnormalities, I said there's 1 million people, at a rough estimate in the UK, with interstitial lung abnormalities, and nowhere near 1 million people with progressive pulmonary fibrosis. There is some debate as to, you know, do all ILAs progress? Some people say yes, radiologically. Do they all progress to disease which is fatal? Definitely not. So there are definitely people who have static fibrosis. By the time they develop symptoms of progressive breathlessness, it's probably not static. Whatever happens to their lung function, it's obviously better. You know, it's a better outcome if the lung function stays stable, but there's still fibrosis going on. But there probably is a large proportion of the population who have fibrosis which will never cause them any problems.
And then they die of something else.
Yeah.
Great. Okay. Thank you very much for a great stimulating discussion. I'd just like to ask Visha to give a vote of thanks.
So, Gisli, thank you very much indeed. First of all, I'm going to thank Gisli for coming to London and to NHLI, which is hugely important, both for the hospital and for the university. So thank you very much. And really, today's lecture was fantastic. Thank you very much indeed. I was actually very impressed just how far ILD has gone. I've read a lot of ILD papers, as you know, in the last 7 years, but this was the best account I've ever heard of where the disease is going. And I somehow feel that one of the most important things we heard today was fibrosis in other organs. And by picking up fibrosis early in other organs, maybe we will be able to detect lung fibrosis at a very early stage. If you'll let me just talk a few minutes about the class of 81, and I'll explain what I'm talking about. So the class of 81, I was an SHO to Margaret Turner Warwick, hugely lucky to be able to sit through 1.5-day ward rounds, Gisli. So it was absolutely amazing. And Lynn Turner-Stokes, Margaret's daughter, will know what I'm talking about because she was a member of the class of 81. This was the SHOs who were together. There were 8 of us. And what was interesting, who else was there in the class of 81? The hugely talented Tim Evans, who sadly has passed away. Pete Openshaw, he's sitting in row number 3 here in the middle, very smugly, and he would tell us all the time that he's going off to do viral immunology. We thought, oh my God, how dull. And that's to the MRC, and he's so brilliant, and how important that has ended up being. And Peter has obviously become a world leader in RSV immunology. We then had Eric Alton, and Eric was telling us, as an SHO, I'm going to cure lung disease with the genes. Then we had Rob Wilson, who was already doing research as an SHO with Peter Cole on infections. So there was this amazing impetus, and Margaret Turner Warwick really inspired us. She was clinically excellent, there was no doubt about it. She was also using science in her day-to-day clinical practice. She was also doing fantastic science, had Pat Haslam doing some of the early BAL work that was ever done. And there were a large number. I was thinking today, driving down from Harefield, I had time to think in the traffic jam today, about all the clinical fellows there were. There was Juliet Miller-Hurst, there was Andrea Penketh, who went to Gloucester. There were others. Margaret Johnson was doing research at the time, who was been at the Royal Free all the time. So there were a lot of women as well here in the Brompton and NHLI at the time. So Margaret was clinically excellent. She used science, and she did science to the very best. She had an interest in biomarkers, all the things that the new centre will take forward. And particularly, she was inspirational to the juniors. She was absolutely amazing, and encouraged us all to go into a new orbit, into a further orbit with respiratory medicine. And all of us I've mentioned took on subjects which were quite tough and maybe not that fashionable. And you know, ILD, as you said, was not at all fashionable. It was an uncurable disease, and it has been made curable. So to finish off, I would like to thank our guests from the Rayne Foundation and from the NHLI Foundation for their generous support. I'd like to thank members of Margaret's family for coming here today. I'd like to thank all the academics who have come, and physicians from other centres, mainly in London. Thank you very much for joining us, to our clinical colleagues from the Brompton, and of course from NHLI and Imperial. And I would really like to wish Gisli, you and the new centre, all the very best. Never, never has there been a better time to have a centre for fibrosing lung disease in the wider sense. We've got COVID now, we're going to get much more pulmonary fibrosis. So what is important is that you use the principles that Margaret Turner Warwick started in order to develop your work and be very successful. So thank you very much for everything. Well done.
So yeah, I think we have
yes,
wine.
Yeah,
so the refectory is open. There is now wine and soft drinks and
I can organise it.
It's okay. So I look forward to chatting afterwards.